Okay, hi, my name is Brad Runsick. Thank you for the introduction, Dr. Newman. I am a county extension agent and have been for about 4.5 years in 2 separate counties in northern Arkansas, and for the better part of that time I've been a graduate student as well, up until about 3 weeks ago. My time split mostly between ag and 4-H, and so finding time for some of these is interesting sometimes. The objectives of this project were really 2-part. One was to determine the presence of plant parasitic nematode species in tall fescue pastures in northern Arkansas. And then from that survey, going forward, to determine the effects of those sting nematodes on the root weights of different cultivars and the suitability of different cultivars as a host for sting nematodes. So the first thing that was done was the survey of tall fescue pastures in the northern tier counties of the state. This was done with the assistance of the county agents in those counties. And so why the survey was done was really just served as a jumping off point for further research. When it was initially started, I wasn't sure what direction we were going yet, but this is where we started anyways. 54 total samples were taken out of those 5 counties from a depth of 4 inches each. And the fescue is known to be a host to at least ring and lesion nematodes. And in this sample, we found several other species that you can see for yourself. Among those, really, root knot and spiral are the 2 that are known to be known to parasitize fescue. Several of the others may, and they may have been there just to parasitize other species that may have been present. Also, should probably note that in those tall fescue pastures, no effort was made to determine what cultivar those were. Those were just existing pastures or the presence of any endophyte. So, the sting nematode, I'm behind. The sting nematode is the nematode I chose to work with. And you may notice from the survey that the sting nematode was not found in those samples. So, the question being, why did we choose sting? For one, like I said, starting the survey, we didn't know what direction I was wanting to go. And when your grad committee chair says, I've got a good population of sting nematodes, let's use those, that's what you do. And so that was a big part of it, but additionally they're a good model. As far as nematode goes, they're fairly large, about 3 millimeters in size if they're full grown. So they're easy to pick out and they're easy to count. And it wasn't known if they were a host on fescue quite yet. The sting nematode's a migratory ectoparasitic feeder, just meaning that it moves around and it feeds external to the root. It uses a stylet to pierce the root, as many ectoparasitic nematodes do, feeding off root tissues. Where it's present, it's one of the most damaging nematodes of the plant parasitic species. It weakens the roots, ultimately resulting in decreased plant vigor and yield. A big note on sting nematodes is that it is most problematic on those soil textures that are greater than 80% sand. So that is important to know when you're. And one of the reasons why we didn't spot those in our survey. Not too many fescue pastures in Arkansas consist of greater than 80% sand, possibly with the exception of some in the Arkansas River Valley. But there may very well be other pastures in the southeastern United States that are grown on a sandy soil such as that. In Arkansas, they're mainly just a problem on golf courses where the sand content is higher. And additionally, they parasitize many fruits and vegetables, other turf species, and other agronomic crops. So what we did, the second part of this, aside from the survey, was to do some host trials and to And to do so, we did 12 replications on each cultivar, these paired comparisons. 6 controls were inoculated and 6 were not inoculated. 6 not inoculated, 6 inoculated with 1 male and 1 female sting nematode. And among those were endophyte infected Kentucky 31, as well as 3 different novels that you can see, and 3 endophyte free cultivars that were used for these checks. Now, past research has indicated that the endophyte infected cultivars may be conferring some resistance to the nematode. So that's in part why I chose these particular cultivars. And the growing conditions, what we did there were 25 seeds of each cultivar sown on the soil surface of 4-inch clay pots. These were greenhouse grown in 300 cubic centimeters of steam pasteurized fine sand to accommodate the sting nematode. They were watered daily. Actually, they were watered 3 times a day. The temperatures were maintained 85 to 90, and they were fertilized with a slow release 14-14-14 osmocote at the rate of 1 gram per pot. That was done 4 times over the course of the experiment. So in order to have some roots to compare against, these were grown for 34 days before being inoculated with the nematodes. Small trenches were dug down into the root zone. The nematodes in water were poured in and covered, and they were left alone for another 64 days. 64 days is nothing special about it, except for it allows enough time for that nematode to cover 2 generations in its life cycle. So it's got time to reproduce twice within that time frame. So the first thing to look at was the reproduction of the sting nematodes in each of these cultivars. And at the conclusion of the experiment, the roots and soil were removed from each pot, and each cultivar was going to be determined to be an excellent host if those nematodes produced at least 8-fold, based on past research. And as you can see, in all cultivars, 6 nematodes had no problem reproducing 8-fold in each cultivar. The letter A indicates if there was any difference, and they all, with the exception of Duramax, endophyte free, they were all well over 1,000. They were all at least 8-fold. Duramax was quite a bit lower, but it was wasn't significantly lower. Also should mention that while they were assumed to be endophyte infected, there was no attempt made to determine if they were indeed or if there were any alkaloids present. So that was an assumption which could lead to some further research. The next component was the dry root weights, and this one is a little trickier. These roots were collected and dried and weighed. The chart shows the orange bars being the inoculated and the green bars being the controls. And that probably doesn't make sense. But in all cases, the inoculated replications were higher than that of the controls. And in 4 cases, in 4 different cultivars, you can see by the letter A, there was a significant difference between the 2. So, actually, I think that's, yeah. Anyway, moving on. So why were the inoculateds higher than the controls? There's been research to show that a nematode infection actually results in compensatory root growth at least for a brief time period. And I believe that that's the response that you're seeing here is in response to the nematode infection, the fescue plant's putting on additional roots to try to make up the difference. So, but I would predict that given a longer duration of infection time to what is ever-increasing population of sting nematodes, these numbers are going to slowly start to reverse. So we're not suggesting that the infection is by any means an asset to the plant. Over time, it's going to catch up with the fescue plant. But again, more work to be done there. So in summary, all cultivars were determined to be hosts for sting nematode, but the study showed no significant difference in reproduction weights between cultivars. And in regard to whether or not the sting infection caused reduced root weights, those results were a little bit inconclusive. Like you saw before, the inoculateds actually resulted in a positive significant difference in a few of the cultivars, but wouldn't predict that that would be the case over a longer duration. Further research to be done in a variety of environments. Of course, these were greenhouse grown, so they were well taken care of. Possibly some outdoor microplot type studies could be done to determine if a particular cultivar provides any greater resistance based on that endophyte status. And for producers managing fescue in sandy soils, proper cultivar selection could be important based on any further research. Especially in my county, there's plenty of fescue pastures that are nearly gone, so the opportunity to plant back with some novels is definitely an opportunity. So with that, I'd like to acknowledge the graduate committee, the nematode diagnostic lab staff, and those county agents, and if you've got any questions, I'll be glad to try to answer them.
Did you actually observe the nematodes in the roots?
No. Well, the question was, did I observe the nematodes in the roots? No, the nematodes are ectoparasitic feeders for one, so they're not entering the root. Well, correct. The stylid is entered into the root briefly to feed, but then they're moving on. So no, this experiment, we did not look at if we witnessed any actual feeding, but they were able to reproduce, so they were feeding on something to be able to get by.
Yes, is there literature showing that this nematode is pathogenic to tall fescue, let's say under a high sand situation or a church situation?
Yes, there is some past research done in the late 70s by Dr. Robbins from the University of Arkansas that did suggest that sting nematodes did parasitize fescue, but at the time that was there were none of these novels to look at, so that was part of why that was chosen.
Also, did you do dry weights of the shoot?
Yes, we clipped them at the soil surface. The dry weights of the shoot was the question. Did we do dry weights of the shoot? Yes, they were clipped at the soil surface and dried for 48 hours.
Were there differences there among the treatments?
Let's see if I can make it work. Yes, they're. Yeah, they were all. They were. What was the question again? Tell me.
Shoot dry weight.
Oh, I'm sorry. No. The shoots were not measured. I misunderstood. The shoots were not measured. Only the roots. So there's lots more opportunity in lots of different aspects of where this could. I mean, it's definitely a very preliminary jumping off point for further, more detailed studies. The question was, what's the initial inoculation rate and the other host for the sting nematode? Again, the initial inoculation rate was 5 females and 1 male. So in all replications, we didn't have any replications that these are just the averages, of course, but in each replication, we had reproduction in all of them. So at least we didn't lose the male, or we had females that were already good to go. The other sting nematodes, many turf species I believe they're a little hard on Bermuda and fescue too, and some vegetable crops as well as some fruits and vegetables. I'm sorry? No legumes. I'm not aware of legumes. I didn't look into that with any of the literature review. Okay. Thank you. Thank you for having me.